Do you use sugar with your coffee or tea? Or do you occasionally drink a sport or soft drink? As millions of people do each day, they obtain energy from the sugar added or contained in these drinks. How can we understand this concept of energy within a sugar molecule? Let's take a tablespoon of sugar and ignite it with a Bunsen burner. You see that lots of energy is released during the combustion of sugar. Water and carbon dioxide are also produced. Obviously, this instant and dramatic release of energy from the combustion of sugar would kill the cell or organism. Therefore, in the cell, sugar is combusted or broken down slowly, in steps rather than instantly, with the subsequent release of usable energy in the form of an energy-carrying molecule, ATP. Click here to learn more about ATP. ATP, or adenosine triphosphate, is the principal energy-carrying molecule in the cell. It is a nucleotide that consists of 3 parts: an adenine base, a ribose sugar, and 3 phosphate groups. Energy is carried in the chemical bonds between the phosphate groups, with the terminal or 3rd phosphate possessing the most energy. Energy is released when the terminal phosphate is cleaved. This energy can be used to fuel the many chemical reactions in the body. When ATP loses one phosphate group, ADP is formed plus inorganic phosphate. The reverse reaction, which produces ATP, ADP plus inorganic phosphate yields ATP, takes place primarily in mitochondria and chloroplasts. Let's see how energy is extracted from a molecule of glucose, a 6-carbon sugar that we know as a monosaccharide. Here is an overview. Glucose is completely broken down into CO2 and H2O during the process of cellular respiration, which includes 3 stages. 1. Glycolysis. 2. The Krebs cycle. And 3. The electron transport chain. Glucose enters this energy-yielding pathway of cellular respiration in the first stage, known as glycolysis. Glycolysis occurs in the cytoplasm, while the Krebs cycle and the electron transport chain reactions occur in the mitochondria. Glucose enters the cell from the bloodstream through a protein transport molecule in the plasma membrane. Once inside the cell, it enters the glycolysis pathway. The first step in glycolysis is the addition of a phosphate group to the number 6 carbon of glucose.
So I set everything up. We're going to do the astronomy.
Select the molecule which will contribute a phosphate group to glucose from the list and drag it to carbon number 6 of glucose. That's right, a molecule of ATP is utilized. Glucose is transformed into glucose 6-phosphate. Energy, in the form of a molecule of ATP, must actually be used to begin the glycolysis pathway. Now add a second phosphate group to carbon number 1 of glucose. Select. Great! You have now primed the glucose molecule by adding 2 phosphate groups. During the addition of these phosphate groups, a slight rearrangement of the sugar occurs, so that the resulting molecule is now fructose 1,6-bisphosphate. 2 ATP molecules have now been used up, so we have a deficit in our ATP account, which will be made up shortly. The next reaction shows us the meaning of glycolysis, or the splitting of glucose. The fructose bisphosphate molecule is split into 2 molecules, each containing 3 carbons as the backbone. Perform this splitting by clicking on the covalent bond. That's right. FBP is split into 2 3-carbon molecules called G3P, or glyceraldehyde 3-phosphate. Notice that the phosphate groups are still attached to the original carbon 1 and carbon 6, which were present in the glucose molecule. The next reaction involves oxidation, or the loss of electrons. High-energy electrons from hydrogen atoms are removed and transferred to an electron carrier molecule. Identify the electron carrier molecule from the list of molecules and drag the correct one onto each G3P molecule. Watch what happens during this oxidation-reduction reaction. That's right. NAD plus is reduced to NADH plus H plus. 2 hydrogen atoms are removed from each G3P molecule. This is the oxidation reaction. NAD plus accepts 2 hydrogen electrons and 1 proton, hydrogen ion or H plus, while a second H plus is left in the aqueous solution of the cytoplasm. This is the reduction reaction. As part of these oxidation-reduction reactions, an inorganic phosphate group is added to each G3P molecule. Choose the correct source of the inorganic phosphate and drag and drop it on the carbon at the end of the molecule that does not have a phosphate attached.
Oops. I was not listening. Oh, crap. Oh, it's water. Oh, shoot.
That's right. Inorganic phosphate is utilized rather than a phosphate group from ATP. Inorganic phosphate is freely available in the cytoplasm. The new molecule now contains 2 phosphate groups and is called 1,3-bisphosphoglycerate. Finally, we are ready to harvest some ATP molecules. 1 phosphate group at a time is harvested from each BPG molecule. This phosphate group is added to a precursor molecule of ATP. Identify this phosphate-accepting molecule and drag it onto one of the phosphate groups. Correct. The phosphate from BPG is attached to ADP, and in the process, ADP is converted to ATP. The resulting molecule is called 3-phosphoglycerate. G3P is rearranged into PEP by the removal of a water molecule and the transfer of the phosphate group to the middle carbon atom. Drag the phosphate group to the middle carbon atom. A molecule of water is released as well during this reaction. In the last energy-yielding reaction of glycolysis, the final phosphate group is removed and a molecule of pyruvate is formed. Identify the phosphate-accepting molecule and drag it onto the remaining phosphate group of PEP. That's correct. ADP accepts the phosphate group and is converted to ATP. From the drop-down menu, choose the total number of ATP molecules that have just been made. Remember that starting with the splitting of FBP, there are 2 PGA molecules that continue. That's right. 4 ATPs have been created, but this is the gross production. How many ATPs were utilized in the early reactions of glycolysis? Choose from the drop-down menu. That's correct. 2 ATPs were initially consumed to prime the glucose molecule. So that results in a net production of only 2 ATPs for each glucose molecule that enters the glycolysis pathway. Most of the energy in glucose is still contained in the 2 pyruvate molecules. Let's summarize our molecular scorecard so far. Arrange the molecules by dropping them into either the glycolysis in column or the glycolysis out column.
Oops.
Pyruvate is the end product of glycolysis. If oxygen is present, pyruvate enters the mitochondrion where further energy-yielding reactions of the Krebs cycle will take place. However, if oxygen is not present, pyruvate will enter a pathway called fermentation. This pathway regenerates NAD+ from the oxidation of NADH. The NAD+ will be used again in the reactions of glycolysis. Let's see how pyruvate is modified in the reactions of fermentation. When we exercise vigorously, our muscle cells may become depleted of their supply of oxygen temporarily. Pyruvate then is reduced to lactate when NADH donates 2 hydrogen electrons and 1 proton. A second proton is picked up from the aqueous solution. Click on NADH to see this oxidation reduction reaction and to follow the path of the hydrogen atoms.
Whoa.
Why is it important to regenerate NAD+? Lactate, or lactic acid, accumulates in the muscles and causes muscle fatigue. Lactic acid is transported out of the muscle and to the liver, where it is converted back to pyruvate. Plants and yeast, a single-celled fungus, have an alternative fermentation pathway that produces ethyl alcohol and carbon dioxide instead of lactic acid. In this two-step reaction, pyruvate loses a carbon atom in the form of carbon dioxide. The resulting molecule is acetaldehyde, which is then reduced by NADH to ethyl alcohol. Click on Start Fermentation to see these reactions. Notice how NAD+ is regenerated again. The brewing and baking industries make use of this type of fermentation, where the release of CO2 causes bread to rise, and alcohol accumulates during fermentation of grape juice to make wine or malt to make beer. Use this tool to construct a concept map of what you have learned about glycolysis. Drag the terms at the right to the correct positions on the diagram.
Fructose. Okay. Oh, first it starts with glucose 6-phosphate. Then it goes to fructose 1,6. Then it splits into Ig3P. And then, oh boy, no pyruvate's at the bottom. 1,3 bisphosphorylation. Something tells me I'm just going to do this here. Yep. And then we're going to cleave it off, but I don't know which one gets cleaved off first. Let's go 3, and then 2. Oh, I'm on a roll. Yeah.
Answer the following questions to review what you have learned about glycolysis. If you get stuck on a question, replay that section to refresh your memory.
Okay. How many ATP are produced per molecule of glucose degraded? So net yield, we'll mix 4, we burn 2, so 2.
You have completed this unit on glycolysis. Good job!